In this video, we will discuss about CAR T cell immunotherapy against pancreatic cancer. Right now, it is just an idea how we can use this CAR T cell immunotherapy against the pancreatic cancer. So, how this CAR T cell therapy works is that first we will take the T cells from the patients who are infected with the pancreatic cancers, and then we can modify these T cells or add the antigens against the tumor, add the antigens of the tumors in these T cells, so that we can modify these uh, T cells, and this CAR is known as chimeric antigen receptor. So this is designed based on the antigen against which we want this immune system to work. So this CAR T chimeric antigen receptors are added or engineered into these T cells. After that, T cells are grown in vitro so that we will have enough population, and then T cells will infused into the patient. In this case, since these are pancreatic cells, it is difficult to infiltrate the microtumor environment of the cells, as it is very dense, and it needs to overcome the immunosuppressive cytokines such as IL-6, TGF-beta, and the dense environment of this fibroblast. So it's difficult, dense environment of this fibroblast and this MDSC cells. So this part is very difficult for this CAR T cell therapy, immunotherapy, to be used against these pancreatic cells. And when we talk about this chimeric antigen receptors, what are the kind of receptors that can be targeted in the case of pancreatic cells are listed here, as this some of them some of the some of these antigens are found in the preclinical and clinical trials in this PDAC cancer patients. So some of the mentions here, such as the CD24, PSCA, CEA, MUC1, mesothelium, FAP, and this HER2. So what is the idea is that first we need to find out whether, which kind of uh, antigens are present in the patients that are infected with pancreatic cells, and then design our this uh, chimeric antigen receptors or engineered T cells that we received from the patients with the chimeric antigen receptors of those antigens that are found in the patients, so that we will have the patient specific CAR T cell immunotherapy. And uh, however, at this point of time, uh, the main bottleneck of this approach is this reinfused CAR T cells into the patients, and they should be able to overcome this immunosuppressive microtumor environment. Otherwise, these engineered CAR T cells will not have that much. Engineered CAR T cells will not have that much impact as in the case of this immunosuppressive tumor environment. The immune system is very weak or unable to act against the cancer cells, cancer or tumor cells, because of the various factors, such as this uh, continuous production of the cytokines related to this IL-6, TGF beta, and this dense environment of this tumor microenvironment will prevent the engineered CAR T cells, CAR T cells uh, to interact with the tumor cells, as we can see here in this in this figure, that CAR T cells, even though they have the receptors, even though they have the uh, antigen receptors against the tumor, they are not able to detect the tumors. So in this case, right now we need to find out a strategy to overcome this immunosuppressive tumor microenvironment, so that this CAR T cells immunotherapy idea can become a reality. If you need to understand about this idea in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.